Welcome everybody to our latest edition of Chestnut Chat. Today we're going to be talking about evolving and emerging pests and diseases of chestnut. We're going to be talking about Gnomoniopsis species, because it's known by many names, with Emily, and we're going to be talking about Asian chestnut gall wasp with Carol. And I've never been good at that scientific name, so I'm going to not attempt that one. It's just ACGW. So as usual, we're in a webinar format. So the only people whose screens you're going to see, who you can hear talk, are the six women who are on your screen. So if you are not on the screen, the six of us, then we can't see you, we can't hear you, you're not going to be able to talk. So go about your day as you wish, and I hope you enjoy our program today. I'm going to go ahead and put a plug in for our August and September programs because I'm excited about those. In August, we're going to be talking about participatory chestnut breeding with Ron Revord and Greg Miller and that program that they're doing out of the University of Missouri. And then in September, I just booked Chuck Ray from Penn State. He's going to be talking about wood properties of chestnuts and some of the results that he's just got. So those should be two fun programs coming up. Let's see. If you have a comment that you'd like to make, please put that in the chat. We've decided that we'd like to stick to kind comments today in honor of Sharon's Be Kind shirt, although we're not seeing her now. Although, yeah, as usual, I'll accept snarky comments as well in the chat. Keep comments in the chat. Do questions and answers in the Q&A. So there's a separate Q&A panel. Please put your questions in there because that's a lot easier for me to keep track of the questions. If you put a question in chat, I might get to it, but it probably also has a really good likelihood of getting lost. We are recording today, so be aware that if you do put something in the chat or in the Q&A, it will be recorded for posterity. Obviously, Emily and Carol, you guys will be recorded today with your presentations. And I think that is all the housekeeping stuff that I need to cover. With that, I'm going to turn it over to Lisa to introduce our latest Chestnut Chat and speakers.
Well, I don't have their bios in front of me, so I'll ask them to introduce themselves when they get ready for their presentation. So thank you, Sarah. These are just a wonderful way for our community to continue to be engaged while we're reemerging from the COVID nightmare that was last year. But everything's going really well at the foundation. I do have one really exciting announcement. Jules, I hope you don't mind, but we been working on a documentary film for about 4 years now, and we've gotten some pretty amazing luminaries to participate, but we just recently this week committed with Dolly Parton to be on our film. So that's pretty incredible. It only took 2 years and going through 6 gatekeepers. I mean, she is a highly controlled and scheduled person, and because she's huge, of course, you know, she's such a well-known and a unifier, and her chestnut story is very authentic. Her Uncle Bill, the uncle that helped launch her career, is a huge chestnut restoration enthusiast, and he sadly passed away last year to Alzheimer's, and I think she's very interested in carrying on his interest and his legacy. We're going to partner with Dollywood in a big way. That's 2.5 million visitors a year to share the chestnut story, and they're interested in educational support for us. So that was a really big win this week. So I just thought I'd share it with our group. With no further ado, however, I will introduce our current stars of Emily and Carol. And who's going first, Sarah?
Emily will. We're talking about. So, and are we calling it brown rot now?
Chestnut brown rot. It's going to be the easiest.
I'm just going to call it chestnut brown rot. So Emily is, like she mentioned, if you weren't here on before, Emily is finishing up her master's degree in plant pathology at Penn State, working on this critter and about to start a doctoral program. What are you going to be working on? Is it potatoes? Am I right about that?
Potatoes, yeah, potato dormancy.
Okay, yeah. So, but I think this is a really fascinating topic, and one of the first, Emily was able to give one of the first documented cases of chestnut brown rot in the U.S., and so we'll cover that first, and then I'll hold most of the questions, if not all of them, until after both Emily and Carol. Carol is at Kutztown University, and she's going to tell us about the northward tracking and expansion of the range of Asian chestnut gall wasp. So we'll talk about that here in about half an hour. With no further ado, Emily, take it away.
All right, thank you. Let me get this going. All right, so hi. As Sarah said, my name is Emily Dobry. I'm a master's student in the College of Agricultural Sciences at Penn State. And my thesis work has primarily focused on investigating and describing domestic isolates of chestnut brown rot and what it really might mean for North American chestnuts and North American hardwood forests as a whole. So today I'm going to be talking to you a little bit about some of the work that we've done investigating that pathogen and give you a little bit of background on the emergence and history of this pathogen. So if you've never heard of it before, you might be wondering what the heck is chestnut brown rot? Well, its scientific name, it actually has two right now, Gnomoniopsis castaneae and its synonym Gnomoniopsis smithogolvii. This is a fungus. It can be an endophyte, so it can live within the plant rather passively, not damaging the plant tissues. It exists in the plant as a saprophyte, meaning it'll live on the dead or decaying tissue. But most importantly, it's primarily known as a pathogen specifically to chestnuts. It's thought to be host genus specific, meaning it's really only thought to be damaging to chestnut species. Now, reports of chestnut brown rot go back as early as the 1980s, but there was a very large increase in incidence of the pathogen in the early 2000s. But the pathogen, the causal agent of that wasn't officially identified until 2012, when it was identified almost simultaneously by two separate researchers, one in Italy and one in Australia, and that's how it ended up with those two separate names. So what should you call it? Well, like I said, those two publications came out really within a few weeks of each other. Gnomoniopsis castaneae is the one that's argued to be the first that was validly published, but if you were to go to Mycobank, which is kind of the leading authority in terms of taxonomy of fungi, it lists both as legitimate, which makes it a little more confusing. So what should you call it as growers? I would suggest that you just stick with chestnut brown rot because that's what's going to be most relevant to you. But I just wanted to give you an idea that it will pop up under these two different names. And actually, castaneae has even changed the spelling a couple times. So you may see it under three names. But the things that are really important for you to know are kind of what kind of symptoms to be looking for. It is known as the primary agent of brown nut rot in chestnuts. And this is a really damaging rot, and it has led to significant crop losses throughout Europe and Oceania. But it is also associated with damaging cankers of chestnut. Although it's thought to be host specific to chestnut, it is found in other woody species. So it's been identified as an endophyte in manna ash, and it has been associated with cankers in hazelnut and common box, and it's also been found in holm oak. So we're seeing that it can be present in more than just the chestnut species. Unfortunately, right now, we don't really know the origin of this. So we don't know whether this is an invasive pathogen or whether it's ubiquitous, meaning it's always been there. Currently, it's believed that it was there all along and we just hadn't identified it, but there is some question as to why we would suddenly see this drastic increase in incidence in the early 2000s. What could have led to that? Potentially the climate change, the global climate change could have contributed to that, but that investigation is ongoing. There are researchers working on that all over the world. So the first official report of this pathogen occurring in the United States was made by some researchers out of Michigan in 2019 when they identified it as the causal agent of nut rot in some of their chestnut orchards there. So this figure is just to give you an idea of how widely spread it is across the globe, and this doesn't even identify all of the locations that it's found. I know it's not the best image, but it gives you an idea of just how big of a problem this is. And if you think about the time frame that this has been detected, first initially described in 2012, and now we're seeing it in multiple states in the US. It's also recently been identified in Chile and Portugal. So we're seeing it globally. Part of the problem with identifying this pathogen is that it's commonly confused with chestnut blight because of those cankers, and sometimes the cankers can look really similar to chestnut blight. So just to give you an example of the way it can present itself differently in different species and on different tissues, I have a few examples here. On the far left, we have the American chestnut, and this is actually from our orchards here. And you can see that the canker is kind of yellow. It's a little different. And then we have an American Chinese hybrid, and that it's a very small canker in this image, but it has the colors that are more reminiscent of chestnut blight, so it's obviously presenting differently than what we see on the American. Next to that, we have a Chinese seedling, and this is from some seedling inoculation tests that are currently underway right now. And as you can see, that's really girdled the tree, and it's kind of more of a brownish red color. So already we're seeing that there's a difference in coloration. And then on the far right is the sweet chestnut, and that was identified in the UK in 2017. So they saw it looking a little differently there. It's almost brownish black. So what you're seeing is that there's quite a bit of variability in how this presents, even within chestnut species. And another problem with this pathogen is that it appears to be systemic, meaning it's found in basically every tissue in the tree. We know that it's found in necrotic galls and green galls. It's found in healthy green leaf tissue, but it's also found in diseased tissue. It's very commonly found in overwintered burrs on the ground, which has been a major issue for vector. And it's also commonly found in the catkin. So we're seeing it all year round and throughout tissues all through the tree. But most importantly, it's found in the chestnuts themselves. And this can be detected as in symptomatic or asymptomatic chestnut tissue. So you might not necessarily see that your chestnut is rotting, but if you were to run a test on it, the pathogen's there. Previously, methods of detection for something like this, and this is pretty common for a lot of plant pathogens, were rather slow. So what you do is you take a sample from your tissue, and from that you try to isolate a fungus or a causal agent, and then you try to, again, you isolate DNA, and then you sequence that, and you try to identify that sequence. So it can be rather time-consuming, a little laborious. It's not very sensitive because you're hoping that what grows out of your tissue happens to be what's causing the problem. But recently, some researchers in Chile did develop a rapid detection method using real-time PCR. And so what this does is it eliminates several of these steps. And what they were able to demonstrate was that you can just take tissue, symptomatic or asymptomatic, you can macerate that up and put in these species-specific primers and go right to the real-time PCR and have a yes or no answer regarding the presence of the pathogen. And this is much more sensitive. So it's able to detect the presence of the pathogen, even if you weren't necessarily able to isolate it in culture. So this is a really promising direction for testing going forward to be able to identify and hopefully mitigate some of the spread of this. Maybe we can get a little bit ahead of this. The primary modes of transmission are believed to be through rain droplets or through wind dispersal, which is pretty common for pathogens of this type. Of course, animals and insects can play a role in that. And one of the insects that plays a really big role right now, or is we know there's an association with, is the Asian chestnut gall wasp. I know that Carol's going to be talking a little bit more about that later. But right now, we don't know whether the Asian chestnut gall wasp actually acts as a vector, as in it's carrying a live fungus from one tree to the next, or if there's just an association where the gall wasp maybe creates the wound and then that gives an opportunity for the pathogen, for the fungus to turn from endophyte to pathogen. We don't know yet. That's also ongoing. But probably one of the biggest methods of transmission is human. Human-mediated transmission is a big problem in terms of plant pathogen because anytime we're going to be sending our chestnut crops overseas, across state lines to sell to market, that can cause us to be transmitting this pathogen without even knowing it's present because, again, it can be asymptomatic within the nuts. We're using grafting materials or shipping seedlings. So certainly we play a big role in the transmission of this. Unfortunately, right now, treatment and prevention for this is relatively limited. In terms of treatment, it's all post-harvest and it's specifically for the nuts. Right now, there was a recent paper describing sterilization in which the nuts are 30 minutes in a 50°C warm bath, and that really helped kill the pathogen so that it was gone and it didn't pose any future issues in terms of storage. There was another research that looked at ozone treatment and found that helped. It allowed for a longer shelf, longer storage life for chestnuts that were infected with this pathogen. So those are the two post-harvest treatments. For prevention, the big one is removing the fall litter. those overwintering burrs are a big one because the pathogen really is in those nuts. If you can remove that, you are certainly decreasing quite a bit of load. And then the other is the sterilization post-harvest to ensure that you're not shipping nuts that are infected. So what does all this mean for United States growers and chestnut orchards? Well, as I said, the first official report of this pathogen was made in 2019. However, in the summer of 2018, we were tasked with observing a small stand of chestnut trees at the Lake Erie Regional Grape Research and Extension Center, which is just an ag extension center of Penn State, and it's located in Erie County, Pennsylvania. They have 15 chestnut trees. They have 5 American, 5 Chinese, and 5 hybrids. So we were tasked with observing these trees for the summer. And while we were there, we noticed that several of them were quite obviously battling some kind of illness or attack. They were sending up shoots from their bases. They had yellowing, wilting leaves. We certainly had an infestation of Asian chestnut gall wasp. And 2 of our Americans had very large cankers on their trunks, which you can see here on the right-hand side of your screen. Now, initially, we assumed that most likely what we were looking at was a case of chestnut blight infection. That's because chestnut blight at the time was really the primary causal agent of damaging cankers, particularly in American chestnut, and that's where we were seeing the most pronounced symptoms within our small chestnut stand. But as you can see here on the left-hand side of your screen is a pretty standard looking chestnut blight canker on a younger tree. It has kind of like that reddish orange or rust color. These tend to be more sunken, they can girdle the trees. But what we were seeing wasn't quite presenting like that. Some of our cankers were splitting the trees. Some of them were kind of a yellowish orange color. So we assumed that most likely what we were looking at was an atypical presentation of this pathogen, chestnut blight. But to confirm our suspicions, we took some samples from those cankers and we isolated fungal cultures. So we followed those really slow methods. And then when we ran DNA analysis of the ITS region, which is kind of it's the fungal barcode region, it's the one that really allows us to identify it down to species. And when we ran and sequenced that, we found that, in fact, our trees were not infected with chestnut blight, or we hadn't isolated chestnut blight, but we had isolated Gnomoniopsis castaneae. So this certainly raised more questions for us. Because we felt like we had almost been duped. We had assumed that what we were looking at was chestnut blight, but obviously that wasn't the case. And we saw that researchers in Europe were having much the same problem. They assumed that they were seeing chestnut blight. They were finding this pathogen instead. And so we made a trip down to University Park to take a look at their chestnut test orchards down there and really get an idea of what chestnut blight looks like on living, growing, actively growing chestnut trees. While we were there, we noticed that several of their trees had symptoms that were really reminiscent of what we had seen here in Erie. And so we took some samples and we again followed those really laborious methods and we isolated the fungal cultures and DNA sequence. And we found that they too had this pathogen present in their orchards, and that's the sample here in the center. And then we were put into contact with Bruce Levine at the University of Maryland, and he had also isolated this pathogen from some of his chestnut orchards there. He'd isolated from Chinese chestnuts, and he was kind enough to provide us with a sample of his isolate. So what you're looking at here on this slide are 3 domestic isolates. So these are 3 U.S. isolates of the same species, but they all look different. They're morphologically distinct. So immediately upon looking at these, you might not think that you're looking at the same pathogen. So this really raised a lot of questions for us in terms of what kind of diversity we were seeing here, if there might be an issue of virulence, one being more virulent than the other. So our primary research objectives for this project were to evaluate the cankering and nut rot potential of these 3 domestic strains, and we wanted to do that in multiple Fagaceae species. So we wanted to look at more than just chestnuts, because we know that out of Europe, they've identified that this has been able to live or be pathogenic, mildly pathogenic within other woody species. So we wanted to look at woody species that would be most closely related to chestnut. So that includes species such as chinkapin, beech, and oak, but we focused primarily on oak, beech, and chestnut for our studies. So to accomplish this, we began with nut rot tests. So in the fall of 2020, we collected acorns from multiple locations around Erie County, And then we were provided with chestnuts harvested from central Pennsylvania chestnut orchards also in the fall of 2020. And we wanted to start with the nut rot test because that's what this pathogen is primarily associated with, is nut rot. For our branch canker tests, we wanted to be able to more fully describe the response of the tree to the presence of this pathogen year-round. So we wanted to look at conditions where maybe the tree is dormant, but if we have favorable conditions for the pathogen to grow and spread, we wanted to see how the tree might respond to that. And so for this, we collected at 3 different times during the year. We collected branches in November of 2020 in the endodormant stage. So this is prior to the completion of the chilling hour requirements of the trees. We collected again in February of 2021 during the ecodormant stage, and that is following the completion of those chilling hour requirements. So this is when the buds, the trees are dormant, but the buds are ready to actively grow if the conditions are favorable for that. And then finally, we collected in April of 2021 during active growth stage. So this is when the trees are beginning to leaf out. And this is when you would expect to see pathogens also start to become more active and grow. So for our nut rot tests, what we did is we surface sterilized all of our nuts, and then we used it's a bone marrow biopsy needle. It's just a little tool to create a wound through the shell and into the soft tissue of the nut. And into that, we placed either one of the 3 live fungal strains, or we placed the control sterile agar. And then we wrapped all the nuts up and we stored them for 1 month. And then we repeated this test. So we completed this test twice. For our oaks, we had northern red, pin, and white oak acorns, and we had 25 of each species per treatment. So that's for each of the fungal treatments and then the control. For the chestnuts, we had American Chinese and American Chinese hybrids, but we only had 11 replicates of each of these, and that's due to a severe drought during the summer of 2020. There simply wasn't a large enough crop to support more replicates for this test, unfortunately, but we were able to complete both of these tests twice. For our branch canker tests, it was pretty similar methods, and we utilized the same 6 species as we had in the nut test, so the red, pin, and white oak, the American Chinese and American Chinese hybrid chestnuts, but then we also included beech. We hadn't included them in the nut infection test because we hadn't had a large enough viable crop, but we did want to be able to describe the beech response to this pathogen in some respect, so we included them in our branch infection tests. And so here again, we just used a tool to create a wound at the midpoint of each branch, and then we put either one of the 3 live fungal isolates, or we put a control, and then we stored those for a month. And each of these was done once for each of those tissue stages, so the endodormant, ecodormant, and active growth. Then to evaluate the results of these tests and kind of the cankering potential or the rot potential, we opened each nut individually and we ranked them on a scale of 0 to 4, with 0 meaning that we didn't observe any kind of rot present on the endosperm or the soft tissue of the nut. But 4 meant that we saw total rot of that nut. It was just completely destroyed. And so here you can see the pictures are kind of demonstrating our sliding scale here, 0 to 4, and kind of how we would rank those. For our cankers, we were able to be a little more sensitive. So we entirely stripped the bark from each branch, and then we measured the length, the widest point and the longest point of each canker. And from there, we calculated the area of that damage as an ellipse. So this allowed us to get a better idea of how big those cankers really were. So I just wanted to give you guys an example of some of what we were seeing as we were taking down these tests before we had even analyzed the data, what we were able to see right off the bat. Across the top here, you have American, Chinese, and hybrid chestnuts, followed by red, pin, and white oak. And in our top row, we have our controlled treatment. And so what you can see here is that there really isn't a whole lot of damage beyond the initial wound site. And that's what you would expect to see. There might be some tissue damage or death, but you wouldn't expect to see it throughout the entire nut tissue. And then in our second row, you can see that's one of our live fungal cultures, and the damage was much more widely spread. And that's what we would expect to see. Unfortunately, particularly in our second round of testing and in our chestnuts, we saw confounding rot in our controls. So we saw that there was rot occurring in our controls, and it in particular looked like brown nut rot, chestnut brown rot. This is likely a result of the presence of the pathogen. As I said, we had already identified that the pathogen was present in central Pennsylvania. That's where one of our strains had come from, and that is where these chestnuts had been harvested. And it was such a meager harvest that our chances of hitting on that pathogen probably were a little elevated. For our branch infection test, we were able to see similar results where that wound would create a little bit of damage, but you wouldn't expect to see that throughout the whole branch. But then in our live fungal treatments down below in the second row, you could see that it was more widely spread, but it did vary by species. So we have the American, Chinese, and hybrid, followed by beech, then red, pin, and white oak. And so you can see that our chestnuts look like they might be a little more susceptible, at least based on these images, but that of course required further analysis. So to analyze this data, it was certainly complicated. We wanted to first test whether treatment was an important predictor of rot. So we wanted to know whether the controls differed from the treated, regardless of strain. And then we also wanted to know whether species was a significant predictor. Did we see that different species were susceptible? And then we wanted to evaluate whether there was a difference in the fungal strain's significance in predicting rot. So we wanted to know if one was more virulent than another. And what we found was that those individual strains were not significant, but treatment and species were. So this allowed us to essentially just pool all of our controls together, pool all of our treated together. We didn't have to consider individual strains. So we would pool control and treated within each tree species. And that's what you'll see in the slides going forward. So for our nut infection tests, both tests produced similar results. So we were able to pool the results of each test into just one general analysis, and that's what you'll see here for our chestnut rot test. Even though we did have a significant amount of rot within our controls in our second round of tests, particularly in the chestnuts, when we pooled that, it didn't change the overall significance of what we found, and the tests agreed. Chinese and hybrid, we did not see that the live pathogen created a significant amount of rot in those nuts, but we did see that it created a significant amount of rot within the American chestnuts. For our oak nut rot test, we saw that all three oak nut species were susceptible to significant rot from the live pathogen when compared to their controls. And this was, this was pretty surprising. We hadn't expected to see this in our oaks. For our branch canker tests, these were 3 individual tests, so we needed to look at them all individually before really kind of trying to describe an overall picture here. And so for our first test, we had the endodormant tissue. That's the tissue that we had collected in November of 2020 before the completion of chilling hours. And what we saw was that red oak and American chestnut were both susceptible to those damaging cankers, the big lesions on those branches, but the rest of the species were not. For our ecodormant tissue, so this is collected in February of 2021, following the completion of those chilling hour requirements, we saw again that red oak and American chestnut were susceptible to damaging cankers, but now we saw that Chinese and hybrid chestnuts were also susceptible to significant damage. And then in the active growth test, we saw the most damage. So we saw a big increase. We saw that the red and pin oaks both were susceptible, and again, all 3 chestnut species. And most importantly, we saw that the Chinese chestnut appeared to be the most susceptible to damage at this stage. We're still in the process of trying to tease out what that might mean and why we see variability here. As you can see, it depends on the tissue that you're looking at and the seasonal tissue and the species. So what does all this mean? What does all this mean in terms of this pathogen in the US and for growers? Well, what we're seeing here is that Gnomoniopsis castaneae, or chestnut brown rot, is associated with significant rot in oaks and American chestnut, so that the nuts of all of those species were susceptible, as well as causing damaging cankers consistently in red oak and American chestnut, regardless of the season. We did see that canker responses differed between those seasonal tissue stages. So what this may mean is that with global climate change and our warming winters, if we have a warm snap, say in December, January, February, where conditions are favorable enough for that pathogen to grow, even though that tree tissue is dormant, we may see that damaging cankers can occur. But we did also see that none of the strains differed in terms of their nut rot or cankering potential. So we didn't see that one was more virulent than another, which is encouraging, at least within this study, within the 3 strains that we evaluated. So the implications for this are, I think they're pretty wide ranging. In terms of host specificity, we've been able to demonstrate that at least in lab conditions, we can say that oak species are certainly very susceptible to this pathogen as well. So it doesn't appear to be host genus specific just to chestnut. It could pose a big risk to oaks. And in terms of ecological risk, that's pretty big. In the eastern United States, we know that following the fall of the American chestnut, oaks moved in to fill the role that they once held. And oaks across the United States are estimated to produce up to 70% of the annual forest nut crop. So they are a major food resource within our forest systems. But if we have a nut rot pathogen damaging that food resource, we could really see that begin to affect those systems downstream, many years down the line. And it could also affect regeneration issues for oaks in our forest systems. But this also could pose a problem for chestnut growers in the fact that oaks might act as reservoirs for this pathogen. Even if it doesn't cause significant losses to their crops, to the forest crop of the oak, this could act as a reservoir to release spores around orchards. And that would certainly require further research in terms of how prevalent this might be in our forest systems. But finally, and probably most importantly for chestnut growers and the American Chestnut Restoration Project, is that what we saw here, at least, was that it's most damaging to the American chestnut nut. And for the American Chestnut Restoration Project, that's a breeding project that really relies on viable nuts. So if we're losing our viable American chestnuts, which already we have a limited genetic pool to include in our crosses and back crosses, this could really certainly complicate the future of the project and the restoration. What we don't know right now is whether transgenics might not have that same problem. That would certainly require further research. So with that, I would like to thank my advisor, Dr. Michael Campbell, and Ivor Knight, who helped fund a lot of this research, as well as Sarah Fitzsimmons and Bruce Levine for providing the chestnuts and one of the strains that were used within this study.
Thanks so much, Emily. That's very interesting, and you know, like we needed something else to worry about. Yeah. You know, I have a grower who she's been growing chestnuts for, I don't know, 20 years, and she said one day, you know, with at the time it was Phytophthora root rot and Gall Wasp, and she's like, ambrosia beetle, which is a terrible thing too. Who has time to worry about chestnut blight? There's all this other stuff that's out there. There are a lot of good questions. I'm going to hold them until after we get, so if you can stick around, Emily, I'm going to hold those questions about brown rot, and we'll talk about Asian chestnut gall wasp real quick, and then come back because we've got some really good questions that I want to get to. All right, Carol, are you ready to go?
Okay. So let me pull up my presentation. Okay, so I'm going to talk to you about the Asian chestnut gall wasp. My background is I got my PhD in plant physiology from Cornell, and I minored in entomology and ecology, and I've been studying galls ever since my graduate work. And at this point, I consider myself a ceccidologist, who is someone who specializes in galls, and in particular insect galls. So I'm going to talk about the Asian chestnut gall wasp. It is Dryacosmus curryphyllis. It's an invasive synipid wasp, and it's of global concern. It forms galls on Castanea species, of course, including the American chestnut. So these are some galls on American chestnuts, and here is the Asian chestnut gall wasp. A little bit of background information. So the Asian chestnut gall wasp is a synipid wasp. There are many different synipid wasps. There are tiny wasps in the family Synnipidae, There are 1,300 or estimated 1,300 species of cynipid wasps worldwide. Most form galls on oaks, some on roses as well, and a few other species. But most gall-forming cynipid wasps are not problematic. In North America, most of them are native and they form galls on native species. Predators and parasites help to control them. Most exhibit an alternation of generations between a sexually reproducing generation of males and females and an asexual generation of all females. So here's an example. This is the woolly catkin gall. It forms in the spring. It's caused by Callioritis quercus operator. Males and females emerge from these galls. They mate, and then the female will lay eggs on acorns. And then you get the acorn pip gall, which is caused by the same species. And so what emerges from these acorn pip galls will just be females. So this alternation of generations is quite common in cynipid wasps. And we'll see how this comes into play for the Asian chestnut gall wasp. So the Asian chestnut gall wasp is not native to North America, and it's considered a global pest. So what are some characteristics of the galls? They're green and or red in color. Here again is a gall on American chestnut. And the galls are unilocular to multilocular. They have one chamber up to 25 chambers, maybe a little bit more. So here's a gall showing some locules, the chambers in the gall. This one you can only see 2 in this particular one. And they're typically from 5 to 20 millimeters in diameter, up to about 30 millimeters in diameter. And this was a very large gall that we found, this one actually on Chinese chestnut. So the life history of the Asian chestnut gall wasp is very important to understand. The larvae actually spend the winter hidden in buds. So they spend the winter in buds. So you might think, overwintering galls contain the larvae of the gall former, but they actually don't. So larvae spend the winter hidden in buds. After they overwinter, the larvae form the galls actually right in the bud area that they were in, and they end up forming galls on developing petioles, leaves, and stems of Castanea species, and this happens in the spring. And so here is a gall forming on the petiole. This is on the midrib of a leaf, and these are again on American chestnuts, and this is on the stem. So this is where you typically find these Asian chestnut gall wasps. The larvae pupate in the galls. This is an early pupa. And then adult female wasps emerge from fully formed galls in late spring or early midsummer. When they emerge is site dependent. It depends on latitude and altitude. So the higher the altitude, they presumably will emerge later. So that is an image of an emerging wasp. And here is one that just recently emerged. It hadn't even completely unfolded its wings. The very interesting thing is there are no male wasps. So this is a cynipid wasp that is females. It's a species that has females only. There is no alternation of generations and no sexual reproduction. The wasps are tiny. They're 2.5 to 3 millimeters long. So adults live from 2 to 10 days. And then the female wasp, and they're all females, will lay unfertilized eggs in Castanea species buds. So when they emerge, basically, they don't have to mate. They just go about laying eggs in buds. And so here are some images of some eggs. They're very tiny. Each female can lay up to 300 eggs. So it takes just one female to start an infestation. Buds can contain 10 to 30 eggs, and they are very tiny, 0.1 to 0.2 millimeters long. So here's an image of some eggs in a chestnut bud, and the eggs are really tiny. Larvae hatch from the eggs in about 30 to 40 days. And here's an image where there's some very tiny larvae in a bud. And then they spend the winter hidden in the buds, forming galls again the next spring. So it's very important to realize that they basically hide in the buds. At the end of the summer through the winter, and they're no longer in the galls. Buds with larvae are visually indistinguishable. So you can't tell by looking at buds whether or not they have the larvae inside of them. If you slice them open, you can potentially find larvae in the buds, but from the exterior, you cannot determine whether or not they contain larvae or eggs. So what kind of damage occurs? Well, inhibited shoot elongation. This image here shows a gall in a terminal position, and there will not be additional shoot growth above this gall. Reduction in flowering and fruiting, nut production, early leaf death and abscission, resulting in leaf area loss. So this is an image showing a gall in this position, and the leaves above it have died. Twig dieback, reduction in winter bud development, reduction in shoot and tree vigor, and it makes trees more vulnerable to diseases, so potentially more vulnerable to the blight. And small trees in particular can die from huge infestations, particularly when there are other stressors present. So the Asian chestnut gall wasp is native to China, and it has spread to many other regions of the world. First spread to Japan in 1941, Korea 1958, and United States actually in 1974. So it's been here for quite a while and it has subsequently spread to other states. Nepal 1999, Italy 2002, and there's been subsequent spread to other European countries. It's considered a major pest in Europe. Canada 2012, Russia 2016. So I'm going to show a map just giving an idea of how the Asian chestnut gall wasp has expanded its range in North America over the decades. So in the 1970s, it was in Georgia, then it spread to Alabama, 1990s into Tennessee and North Carolina, and then the 2000s that had spread to many other states, and the 2010s even more so. And it's also now in Canada, as I had mentioned previously. Its range pretty much overlaps with the native range of the American chestnut. Now this map here does not show distribution within individual states, but I've indicated which states have the Asian chestnut gall wasp, or at least which states have recorded the presence of the Asian chestnut gall wasp. So how does the Asian chestnut gall wasp get dispersed? Natural means, by active flight. However, the gall wasp is not a strong flyer. It's induced to fly at low wind speeds, and then it's carried by the wind. It's blown by the wind at very high wind speeds. And so the direction of prevailing winds is really important in the spread of the Asian chestnut gall wasp. In terms of rates of dispersal, so short dispersal distances and rates have been estimated to be 3 to 18 kilometers a year, which is about 2 to 11 miles per year. Historical dispersal in the United States has been estimated to be about 15 to 25 kilometers a year, 9 to 15.5 miles a year. Unnatural means of dispersal would be human transport of infested plant materials, such as infested seedlings, saplings, or scion wood for grafting. So looking at this map again, I've added some yellow spots where the dispersal is suspected to have occurred by unnatural means. So humans transporting infested plant material. Well, actually, I should have put a spot on Georgia because the initial introduction was actually by someone bringing the pest over inadvertently, but by unnatural means, obviously. So, so some of the more recent dispersal of this pest seems to have been due to this unnatural dispersal, as opposed to the early range expansion is thought to have occurred more due to wind and flight of the gall wasp. So this is a map showing the range of the Asian chestnut gall wasp. It was last updated March 2019 and shows different counties where the gall wasp was found. And we did some work to survey sites to see whether we could find the Asian chestnut gall wasp in other locations. And so this map here shows, actually it correlates with the counties that were on this map. They're shown in gray. And then we visited many sites. So the green spots are areas we went to where we surveyed chestnuts and did not find the gall wasp. And the red spots are areas we went to where we surveyed chestnuts and found the gall wasp. And so as a result of our surveys, we basically extended the range in terms of documented the extended range into New York, New Jersey, and Delaware, adding those 3 states, and then 27 additional counties we've added to the range of the Asian chestnut gall wasp. So we wanted to look at some of the factors that might have affected the dispersal of the Asian chestnut gall wasp. So I'm not going to go into a lot of detail here, but I'm showing the map from 2019, but we had visited some of these sites earlier in that decade, and then got an idea of dispersal rates and an idea of where and when the wasp was dispersing. So the dispersal into Delaware presumably resulted from nearby populations. Actually, you can see Delaware County in Pennsylvania already had the wasp, as did York and Frederick in Maryland. So it's easily explainable that the wasp ended up in Delaware. New Jersey, very close to Pennsylvania. So infestation presumably came from Pennsylvania. This southwestern population in New York, southwestern New York, presumably ultimately came from this site in Ohio. There were intervening counties here in Pennsylvania that had infestations. There are probably more counties here that were infested, but just weren't recorded. We did find the Asian chestnut gall wasp in south central New York. And so, I was very interested in trying to get an idea of how it got there. So it could have gotten there from Pennsylvania in terms of the speed of dispersal. But in actuality, when we visited these sites in northeastern Pennsylvania, we didn't find the gall wasp. So in order for it to have dispersed naturally from Pennsylvania going north, we would expect to find some stepping stones here where we would have found infested trees, and we didn't find them. And so it doesn't look like it came from southeastern Pennsylvania. And as far as coming from the west, actually the dispersal rates would have had to be much more rapid than has been the case historically. So it really seems as though this infestation was due to introduction of contaminated plant material. In addition, we looked at some of the topographical features just to get an understanding of the dispersal of the wasp. And it seemed as though the Ridge and Valley province in Pennsylvania, which is shown in pink here, basically seemed to inhibit the northern movement of the wasp. So actually there's some sites here. We visited these sites and did not find the wasp as of 2019. And there were just a few places, and it basically seemed as if the wasp was actually following the Susquehanna River, and more specifically being dispersed along the Susquehanna River Valley through this Ridge and Valley province. Another thing we looked at was the highways running through here. It's been hypothesized that these wasps might hitch a ride on vehicles, but we did not see evidence that the north-south highway, in particular 81 running through eastern Pennsylvania, if you look at the sites along here, we didn't find infestation, even though the highway runs through here. We also found some evidence for enhanced dispersal along the Great Valley. And basically this Great Valley area showed a lot of infestation, and the winds come from the southwest during the prevailing winds come from the southwest during the dispersal time of the adults. And so that could easily have helped spread the wasps. So in our surveys, we found that these wasps were found primarily on Chinese chestnuts, on residential or business properties. Sometimes those business properties were old residential properties that were converted to businesses, or old farms that were converted. We also found galls on chestnuts in botanical gardens, orchards, on campuses, and we did find the gall wasp on American chestnut saplings. But there are many areas where you can find Chinese chestnuts in Pennsylvania in particular, and those Chinese chestnuts are acting as reservoirs for the Asian chestnut gall wasp. And I found that most people, you know, we went to over 100 sites when we surveyed for the wasp, and most people didn't know anything about the Asian chestnut gall wasp. So I think there's really a need for much more awareness of this invasive species. So how can one control the Asian chestnut gall wasp? Pesticides. Insecticides have not been shown to be effective against the Asian chestnut gall wasp. The larvae are well protected in the galls early in the season. And then the bud scales provide protection of developing larvae in the buds later in the season. So remember, they overwinter in buds. And then one could potentially use pesticides against emerging adults, but you need to know when the adults are emerging. And that really varies from place to place. So timing is very important, and you potentially need to use very large quantities of insecticides. There are toxicity concerns and environmental concerns, such as negative effects on pollinators as well. So pesticides do not seem to be the solution. Developing resistant American chestnut hybrids. Ozark chinkapins are rarely galled. Allegheny chinkapins show resistance, and also Chinese chinkapins to a certain extent. So there have been some limited attempts to do some crosses, basically Ozark chinkapin times Chinese chestnut hybrids. Actually, American chestnuts crossed with Ozark chinkapin Chinese chestnut hybrids. And after 14 years, these trees, the ones that survived, some of them had no galls. So there needs to be more research to see if American chestnut hybrids can be developed that are resistant to the Asian chestnut gall wasp. So in the 1970s and the late 1970s, several parasitic wasps were introduced into Georgia. And one of them was Torimus sinensis. Torimus sinensis is native to China. It's now established in Japan, Europe, and North America. It's a parasitoid. It actually ultimately kills the host, and that's why it's considered a parasitoid. Biocontrol with Torimus sinensis seems to be the most effective method for controlling the Asian chestnut gall wasp. So this parasitoid will control levels of the Asian chestnut gall wasp, but will not obviously eradicate it. So here's an adult, and there's a larva, and this adult was reared from an American chestnut gall, and this was the larva inside an American chestnut gall. So remember, these stay inside overwintering galls. So in terms of the life cycle, the adults emerge from those overwintering galls in the spring. So here's an image of an adult Torimus sinensis. And after mating, the female lays eggs on the larvae in developing galls. The female has a very, very long ovipositor that enables her to lay the eggs inside the galls. And each female can lay an average of 70 eggs. And then the larva will feed on the Trichosmus curryphyllis larva, eventually killing it. And then, as I've stressed, larvae of Torimus sinensis overwinter in the Asian chestnut gall wasp galls and then will eventually pupate. And here's a larva, again, an image of a larva. Adults emerge in the spring, and sometimes actually they will emerge in the subsequent spring. Other attempts for controlling the Asian chestnut gall wasp would be hot water treatment of dormant buds. And this actually has been studied on Chinese chestnut scions, as well as European chestnut scions, treating the scions in water in both of these instances 10 minutes, but the temperature is different. And so one really needs to look at this closely for particular species and varieties. And these are two studies that showed success. They were able to kill the larvae in the buds and then have successful grafts, but one needs precise temperature control and timing for this to be successful. So more research is needed on hot water treatment of other Castanea species, such as the American chestnut, as well as hot water treatment of seedlings. Surveillance and pruning. So pruning of branches with newly formed galls in the spring before the adults emerge is one way to treat for the Asian chestnut gall wasp on a small scale. And then one would want to dispose of the galls in a manner that kills the wasp larvae or whatever stage they're in at that point. So burning is a possibility. Removal of galls, however, after the gall former has left, will result in removal of the parasitoid Torimus sinensis that overwinters in the gall. And so whether or not one wants to remove those is really dependent on other factors, such as whether or not those galls are actually harboring fungi, such as the blight fungus. So, but if you do remove galls later in the season, you are removing the parasitoid Torimus sinensis that will control Trichosmus curryphyllis. So some emerging concerns here. Heavy infestations of the Asian chestnut gall wasp may make European chestnut trees more susceptible to blight. And the blight fungus has been cultured from abandoned galls. The fungus may actually enter the galls through the wasp exit holes on abandoned galls. In addition, there have been studies that, and this here is a gall that has blight symptoms, and they basically were able to culture the blight fungus from the galls, but also, as Emily had mentioned, the brown rot fungus has also been found in galls. So the plot thickens, basically. And one additional emerging concern is there is a new gall former, Trichosmus zuili, and it forms galls on Castanea. It was described in 2015, found in southeastern China. So it's in the same genus as the Asian chestnut gall wasp. It forms galls that look just like Asian chestnut gall wasp galls. So far, it's only been found on Chinese chinkapin, but it has not been studied extensively. And what's really fascinating from a biological point of view is it actually exhibits sexual reproduction. There are male and female wasps. So given that it reproduces sexually, and given that there is potentially more genetic variation as a result of that, this could lead to greater adaptability, making management more difficult if this becomes a global pest as well. So to date, it has not been reported outside of China. So more awareness, more research is needed to help curb the spread and impact of the Asian chestnut gall wasp on the American chestnut and other Castanea species in North America. And I'd like to acknowledge students and colleagues who have helped me, as well as my husband, and the property owners, managers who allowed us to observe and monitor chestnut trees for the Asian chestnut gall wasp. Here are some references, and that's the end.
always hate to say excellent after these these presentations about these pests and pathogens, but that was a very good presentation. Thank you, Carol. We have lots of really good questions. I'm going to take the, because I'm the moderator, I'm going to ask the first question. Carol, I have a question about infestation of trees in a cultivated environment, and that doesn't mean non-native, but I guess in a cultivated environment versus wild. What do you see less chestnut gall wasp in the forest than you do in cultivated settings, and if so, why?
I think that's a great question. So I personally really, one of the things that was driving me is I had been monitoring the Asian chestnut gall wasp in Tompkins County, New York, and it wasn't there for many years, and then it showed up one year and I thought, how did it get there? My immediate thought was it came from Pennsylvania. So my idea was to get an idea of how it has spread in the eastern part of Pennsylvania. So I have not really looked at that many natural sites. Actually, I focused more on Chinese chestnuts. But for natural sites, it's kind of interesting. I have found them on saplings. There was one site that was fairly heavily infested, whereas there's another site that I went to that is near sites that are infested, but it seems to be kind of protected in the I think actually the wasps have a harder time getting to some of the natural sites than they do hopping from one Chinese chestnut to another. And you know, I haven't studied this specifically, but it's my feeling that that might be the case.
That's my observation too. And then here, you know, here in State College, we've had gall wasp since at least 2014, maybe before, but that was the first time that I've noticed it in our orchards at Penn State. But then again, you know, we've got orchards in the mountains that have yet to be infested, even though there are infestations nearby. So it is kind of odd the way it kind of hops around. But it is my observation, like yours, that large Chinese chestnuts or large orchards tend to be more attractive, and that makes sense, you know, than.
Yes. And I was really interested in this Ridge and Valley province situation because I noticed when we go up in the mountains, I would find a lot of chestnut trees, Chinese chestnut trees that weren't infested. So, you know, I think there's something to that as well in terms of dispersal. So, and another thing is abandoned orchards are a problem. So I found some abandoned orchards that were heavily infested. So
Abandoned Chinese chestnut orchards.
Chinese chestnut orchards, you know, other chestnuts as well, but yeah. And they really harbor a lot of these gall wasps. So
do you want, so I've got a couple people on here. Mike Alcott says he sees them in Mercer County Park in Trenton. I'm from West Virginia. I've seen them, you know, all over the place there. Do you want people to report these locations to you? Are you interested in following this, or?
yeah, sure, yeah, that would be cool. So I have my email address on my slide. I can just put that up again. And I'm actually, one of the other things I'm going to do is I'm going to go back to the sites that up in northeastern Pennsylvania where I didn't find the wasp, and I'd like to find out, you know, when it gets there. So, and there's actually a controversy in terms of the speed of dispersal. In Europe, it seems as though it's not moving quite as quickly as we have thought it was moving in North America. So, you know, and we're in a different situation here in terms of, you know, where the chestnuts are, et cetera. So they have chestnuts in forests and they're larger. And so it's really quite interesting. So I'll put up my PowerPoint again with my email address. Actually, I need to share the screen.
Do you know if there's any association of these, or if they seem to prefer infected trees?
And when you say infected trees,
so typically people would be looking for blight in chestnut. So are we seeing that trees that already have blight are more likely to have a bad infestation of chestnut gall wasp?
I'm not sure that's the case. You know, I've seen heavily infested trees that didn't look like they had blight. So, but you know, this whole interaction with blight and other fungi, I mean, this is a huge area of potential research because different fungi have been cultured from the galls. So, and actually, I found one study where they thought that the brown rot fungus actually might be killing the larvae in the galls. So, you know, it's not clear what is going on.
Yeah.
So it's very fascinating.
Well, good. Well, so thanks, Carol. So any of you who have a report of a location, see Carol's email address there, mapes@kutztown.edu. Frank, he says he's got gall wasps in Tuscaroras County in Ohio, and I'm sure we could get a pretty good complete map of where these things are. I'll take Anthony Cicilli's question first. What would using a microwave kill the eggs?
Well, it might also kill the buds too. So I don't know if anybody's looked into that. I've actually had students use microwaves in my plant physiology class to see whether they, can affect growth of plants, etc. But it doesn't take much to kill buds. So, but that's a good question.
Yeah. Anonymous question, wouldn't the gall wasp be a good thing for controlling other problematic insects?
Would the gall wasp be good for controlling other quick answer is I don't think so. So that wasp actually, it doesn't interact with other insects. So I'm going to stop my screen share here, but it's not really interacting with other insects. Now, there are some native predators and parasitoids that have been found in Asian chestnut gall wasps, galls. And so, but really, they do not seem to be inhabiting or affecting Asian chestnut gall wasps that much. So, Torimus sinensis is really quite effective, but personally, I wouldn't put all my eggs in one basket, basically. So.
It's my observation that it gets worse before it gets better. Our infestations across Pennsylvania and our orchards, and then our Meadowview research farms for TACF in Southwestern Virginia, you kind of have this slow burn for a couple years, and then like it's just everywhere, and you're like, I never want to grow chestnuts ever again because this just looks so awful. And then the parasitoid finally takes control. So you've got like this 5-year period of kind of up, huge infestation, and then the parasitoid takes control. And then, we've got it. Like 2018 was terrible for us. Here we are, 2021. It's there. You see galls, but it's not horrible. And that seems to be the case. I mean, I think our Meadowview research farms have had gall wasps since 05, 06, and it just seems to be a fact of life. Now, if I were a chestnut commercial grower, I would be much more worried because that affects your livelihood.
Right. And I think there needs to be more control, over growers and, what's being sold, etc. So.
Sure. Yeah. To keep it from getting around. All right, we got tons of questions about chestnut brown rot, Emily. A lot of people are asking about the transgenic tree, and you did mention that. Do we know anything about the possible effects of the OXO gene or other transgenic trees on chestnut brown rot?
Right now, as far as I'm aware, nobody else in the US is really looking, particularly in terms of American chestnut. And that's what the transgenic is the American chestnut. But a lot of these canker-causing fungi use oxalic acid. That's how they create that damage. They break down the tissue for them to be able to metabolize it. So it is likely that this pathogen uses oxalic acid, but we don't yet know. We're currently in the process of sequencing the genome, so hopefully we will have some answers to that at some point, whether this is present, whether it's being utilized. But I would love to see researchers out of SUNY take this pathogen, take my pathogen, and do something with it and see if they can find out if they might be at risk. I think probably the transgenics will be in good shape. That would be my suspicion.
Is chestnut brown rot a necrotroph?
Yes.
Yeah. Okay. Michael Hagan asks, can brown rot cankers be treated with mud packs like chestnut blight?
I have not seen anybody try it. I have not seen any mention of it being tested.
How lethal is chestnut brown rot fungus compared to chestnut blight? Does it typically girdle a tree?
That again depends on the tree species that you're looking at. So most of this, almost all of the studies up until ours, nobody else had looked at Chinese or American. And that's primarily what we focused on because that's what hybrids really are here that we want to protect for the American chestnuts. But in terms of the sweet chestnut hybridized with the Japanese chestnut, it doesn't look right now like it's lethal, but it is certainly, it's not as aggressive as chestnut blight. So it might be a slower growing pathogen. It may eventually be lethal, but you might have more years out of your tree. But the problem is you lose a lot of your crop.
Right.
So you might lose future generations.
Anonymous asks, what temperature were test subjects stored at and why? And I think that's for the nut storage.
For the nut and branch infection assays, we stored them at 25 degrees Celsius. That's the between, I think it was 23 and like 27 degrees Celsius is considered the sweet spot for this pathogen to grow. And we had a 25C, so we stuck it smack in the middle of that sweet spot.
So with brown rot in commercial crops, this is my question. Typical storage conditions for chestnut is 0 or 1C, well, 1 or 2C, because you want to try and keep it fresher longer. What are chestnut orchardists seeing in the commercial industry as far as damage and crop loss due to this? Do you have any sense of that?
From what I understand from overseas, in terms, I don't really know much about the U.S. in terms of their crops, but I know that overseas what they're seeing is that a lot of times what's happening is when they're going through their sterilization process, part of that 30C warm bath was finding the way to kill it so that it wasn't transmitted to other nuts, that as you store the nuts, that pathogen can't take over. You're looking at a very small, the nuts are small, particularly when you're looking at the sweet chestnut, right? It's a small tissue, it's a very soft tissue, it's very high in a lot of the stuff that a fungus wants to grow. So even at those colder conditions, it seems to be causing damage and a lot of loss within a very short amount of time. Within probably a month, I think, of storage, they're losing huge amounts of their product.
Wow. So a hot water, because I know a hot water bath is often used to treat chestnut weevil, and it seems like that's going to be another thing to use for treatment of brown rot as well.
Yeah, I can send you the link if you want to share that to this recent paper. I think it was just published in May, where they had done some studies over what was the best temperature to really kill this pathogen and prevent it from rotting your nuts and storage.
Spreading. Yeah.
Yeah.
Let's see. Cable Smith asks, were the red oaks less affected by the pathogen because they usually grow in a genetically diverse environment? For example, lots of different species in a forest. I'm not a scientist. Whereas chestnuts are farmed and grown in a genetically homogeneous environment.
That's certainly plausible. Most likely what's occurring is that this pathogen started in chestnut, that it really is primarily associated with chestnut for a reason, but that what is it, sudden oak death that can be found and cause damage in other species, that we're seeing that this oaks, which are very closely related, they're closer than beech, they're also susceptible. And two of the most closely related species of Gnomoniopsis are pathogens in oak. So there's probably already that pathway there for a Gnomoniopsis species.
Let's go back to Gall Wasp. Carl Absher. Hi, Carl. Have you tried systemic insecticides on Gall Wasps?
Sorry. So I personally haven't, but there have been studies where people have used systemic insecticides and they haven't been particularly effective. So insecticides aren't particularly effective against Gall wasps in general. So.
Yeah. Jenna Meyer, I have galls on my plum trees, possibly Phomopsis. My chestnuts are small. Can that transfer to the chestnuts?
Not sure. Do you know, Emily?
I'm sorry, I was reading something in the chat. What was the question?
Do you know if Phomopsis can transfer from plum trees onto chestnut?
I have no idea. My focus has been Gnomoniopsis. I'm sorry, I'm not widely versed in fungi.
Frank. Hi, Frank. Last summer, I don't think this is about either. This is sort of a general question. Last summer, I had 3 chestnuts that had grown well for several years and were about 6 to 7 foot tall. Looked really healthy last summer, fully leafed out, growing July, August. All the leaves died on these trees. 2 had small root sprouts. This year, they have no sign of life. Root sprouts died too. A forester told me likely a fungus since the wet spring. Is this possible? How susceptible are chestnuts to wet weather? Maybe. And so I don't know if I can drag up a picture really quick, but I recently had Mark Double, who I know is on this chat. Hi, Mark. Had sent me a picture of a grower, a Chinese chestnut grower and hybrid chestnut grower in northwestern Pennsylvania. And same thing, tree just up and died. Like vascular cambium looks fine. Leaves are just like a dark brown, which that's not typical of chestnut blight or even Phytophthora root rot. It's really perplexing. Hill Craddock always said when sometimes chestnuts and trees in general, but chestnuts just die for no good reason. And I always thought that was an excellent way to describe it. No good reason. Wet weather, I think, does affect health overall. I mean, there are lots of pathogens that prefer wet environments and can certainly affect chestnuts, and chestnuts don't like wet feet. And so depending on where they're growing, it could certainly affect, their overall health and mortality. But, specifically, I don't know that we know. Carol or Emily, any thoughts on that?
My first guess would have been Phytophthora, but I'm not extremely familiar with it, but I know that would be a ground up, a base up kind of death, especially since it's an oomycete, right? So that wet weather would certainly be favorable to it.
If it's there. So I know Frank's in east central Ohio, where I don't think it's been seen yet, but that doesn't mean it's not there. But it would be worth testing, I think. Dan, has anybody explored the connection between micronutrient nutrition and brown rot? The massive increase in EMFs may be affecting new and emerging. I don't know what EMF is. What's EMF?
Like electromagnetic field? What is EMF?
Oh, massive increase in EMF. Dan, can you tell us what EMF is? May be affecting new and emerging diseases by affecting plant immune systems as it does human immune systems. Any thoughts?
That is a very good question.
Let's talk about micronutrients and brown rot. I mean, is there any sense of I think with anything, the healthier the tree, the more robust it is, the more it can fight off infection. Are you seeing any indication of that for brown rot?
I don't know that there's been a whole lot of work looking at any kind of nutrients. This pathogen was only described in 2012, and a lot of it has focused really just on the nut rot and identifying cankers. So there hasn't been, unfortunately, a lot of in-depth work with this pathogen.
And Carol, I'm going to ask the same question to you. So Dan says yes, electromagnetic frequency. But Carol, we kind of got this with, does chestnut gall wasp infect trees that have blight? But I am curious, were you able to define any difference in infestation versus overall health of the tree? I know you can't. How can you tell, is it a chicken or an egg? Like, was the tree not healthy and therefore became infested, or now it's infested and therefore it's unhealthy? Like, I do see that blight tends to do a lot more damage on trees that are infested with gall wasp. Can we tear those apart? Have you noticed that in any of your observations?
Yeah, I think it's a little bit hard to tear because it's the chicken and egg situation, in terms of why is there an association between the blight and the galls, actually? So that's one of the questions, basically. I mean, are the galls actually causing more blight symptoms too? So that really needs to be studied more. Yeah,
that's my observation, but yeah, I don't have any quantification of it. Russell says, hi Russell, have you tried to take some blight from a tree, put it in a bag, look at it with a microscope, and then make a spray, put in the bag and see if it kills the blight? I have not, and I have not put blight in a bag. I do have blight growing on petri dishes. I've never sprayed them with anything. I know some people who are looking at ways to control blight do that. You know, they throw on alcohol or some bacteria or whatever and see if that helps control. And, you know, so when Mark Double gave our chestnut chat in April, we talked a lot about other possible controls of chestnut blight and how sometimes it works on a petri dish and it works in the lab. And then when you go out into the field and apply it, it doesn't. But I still think that it's worthwhile to see what all can kill blight in a petri dish. I think that is interesting. But then to replicate it.
Getting back to your question, Sarah, I know about other galls. I mean, I go looking for galls all the time, not just on chestnuts. And I've often found that it's healthier trees that tend to have galls.
Oh, okay.
Yeah, so I don't know though with the chestnut gall wasp, but I've often found that unhealthy looking trees don't tend to have a lot of galls on them, particularly oak trees. So,
but that would make sense, right? I mean, the wasp would want something that's healthy, that it can rear, that it's ensuring that it's going to rear its young on, right? So, not that they're sentient. Are they sentient? Does that make sense? They're becoming self-aware. So I got one more question and we're about to wrap it up anyway. Anonymous asks, does bacteria coincide with the fungus? And so Emily, I think that's for you. Do you see bacteria hanging out with brown rot?
Well, typically when you isolate, you're looking for one or the other. So we haven't looked for any bacteria. I know that there have been studies in Europe that found that certain bacteria are antagonistic to the fungus, so they will limit its growth. And there's ongoing research with that as well to try and determine whether there might be ways to apply certain treatments that could be antagonistic. I also know that chestnut blight and brown rot are almost never found in the same tissue. So if you have chestnut blight in your trunk, you're not going to have brown rot there, but you might have brown rot in your leaves and nuts and won't have chestnut blight there.
Well, that's interesting.
Yeah, they don't know why.
Yeah. You grew them together, didn't you? At some point,
I tried growing them on a plate, and we never isolated chestnut blight here. We tried growing the 3 strains together to see if we could get anastomosis. It was Bruce Levine that looked to see if they were antagonistic, and he didn't see anything on a plate. They just avoid each other.
They hang out just fine together?
They just avoid each other. They don't anastomose. They don't bother each other. They just both grow and then they stop.
You do your thing, I'll do mine.
Yeah, pretty much.
We'll kill this tree together. Let's do what we can to kill this tree and eat it.
We'll just divide it up.
Yeah. Well, good. Well, thank you guys so much for joining us. I really appreciate it. So we'll wrap up here. So for everybody, thank you for coming for our latest edition. Like I said, in August, we're going to have Ron Rivord from the University of Missouri, joined by Greg Miller, who heads up the Route 9 Chestnut Cooperative out of Ohio. And then in September, we're going to talk about wood properties of chestnut with Chuck Ray. With that, I'll say adieu and turn it over to La Grande Fromage.
Madame Chestnut. Thank you, Carol and Emily. That was excellent, although it's more things that we need to worry about, right? But I'm glad you're on the case and helping us out get this tree back in the forest. And thank you, Sarah, for your excellent facilitation. I had to do the last months by myself, and I realized how much work and gymnastics it takes to manage these, but you did a great job as usual. And thanks to all the participants. Have a great weekend. Thank you for all you do for TACF, both in the field, in your pocketbook, and everything else and above. So everybody have a great weekend. Thanks again, Carol and Emily.
Thanks. Thank you.
Thank
Nice
you.
job, Emily. Thanks.
Thank you. You too.